C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with lipidomics. In today's session, we will start with overview of metabolism. Experimental study of metabolism. The study of metabolic sequences can be conducted at 6 levels of organization. Level 1, the intact organisms. The essential nature of amino acids and vitamins, etc., could be understood by feeding animals with diets lacking in one of the ingredients of food. Radio-labeled iron is given and incorporation of the radioactivity in bone marrow and erythrocyte precursors are studied, which provide information regarding the lifespan of the RBCs and the rate at which heme is degraded. The studies of inborn errors of metabolism is helping in understanding the normal processes inside the body. This is the diagram of radio-labeled iron, which is fed to the rat. After that, it shows that there is decreased organ distribution and decreased half blood life. It is easy to study individual enzyme systems in microorganisms. By utilizing mutant strains of bacteria, metabolic defects may be elucidated. Coming to the next, organ perfusion. The organ can be isolated, preserving its blood vessels. The organ is cannulated and perfused with ringer solution. To the perfusion fluid, any compound may be added and the fluid emerging from the organ is analyzed for the metabolites of the compound. Coming to level 3, organ slices. The next low level of study is used by slices or slices of organs about 50 micrometers thick. The organs of this procedure is that cellular organelles were preserved intact. Metabolic transformations of nutrients can be studied in detail. If rat liver slices are incubated with medium containing glucose, the carbon dioxide is evolved. Level 4, intact cells and tissue culture setup. Tissues or cells can be kept in defined culture medium, for a few days for metabolic studies. The medium contains nucleic acids, carbohydrates, amino acids, vitamins, and growth factors. The pH of the medium should be kept around 7.2. If labeled glucose is added to the culture, the utilization of glucose and its incorporation into glycogen, etc., could be identified. If the labeled nucleotides are added in the culture, the cells take them up for DNA synthesis, and the uptake of radioactivity will be proportional to the cell division. In this diagram, we can see that there is radio-label nucleotide which contain gamma 32p GTP as a radio-label nucleotide. Advanced activities of drugs can be studied in the relation cell culture system. The biologically useful substances can be harvested and tissue culture set up. For example, specific monoclonal antibodies could be obtained from the supernatant culture of hybridoma cells. In this diagram, there is mouse which is challenged with the antigen, and from mouse spleen cells are extracted. Then the spleen cells are fused with myeloma cells to form hybridoma cells, which later harvest monoclonal antibodies. The cells from the cancer tissues have indefinite capacity to grow into a number of passages. The immortalization is characteristic of cancer tissues. The good example is HeLa cell line from cervical cancer tissue now growing in laboratories all over the world. Level 5 Homogenates. The tissue is homogenized in an isotonic medium and cell wall is broken by ultrasonic vibration and cellular organelles are separated. For example, isolated mitochondrial preparation will show enzymes of electron transport chain. Level 6A Purified enzymes. Enzyme preparations may be used to study individual metabolic reactions, their regulation, and cofactors, etc. Level 6B DNA or genomics. Present day research work the mainly involves the studies at genetic levels or molecular biology. For example, phenylketonuria is due to a mutation in the gene coding for the enzyme phenylalanine hydroxylase. The complete the full complement of genes within the cells are known as genomics. Their expression and regulation is known as transcriptomics. The gene products, that is the proteomics, can be thus studied. Coming to the use of radioisotope tracers. The isotope studies provide valuable information regarding precursors, which include product relationship, rate of metabolism, anatomical distribution, etc. When C14 labeled glucose is administered, the metabolites can be traced to different organs. Administration of N5 labeled glycine, which was followed by appearance of the label in different compounds like hemoproteins, nucleic acids, and creatinine. Coming to studies on metabolism, four aspects of metabolic pathways are studied: sequence of reactions, precursor product relationship, mechanism of reaction, and control mechanisms. Metabolic pathways may also be studied by creating disturbances to the system, such as by causing metabolic blocks, by studying organisms with metabolic defects, then gene manipulation, example, gene knockouts, etc. Coming to metabolism, thousands of chemical reactions are taking place inside a cell in an organized, well coordinated, and purposeful manner. All these reactions are collectively called as metabolism. The metabolism serves the following purposes. First, chemical energy is obtained from the degradation of energy rich nutrients. 2. The food material is converted into the building block precursors of cellular macromolecules. These building blocks are later made into macromolecules such as proteins, nucleic acids, polysaccharides, etc. Biomolecules required for specialized function of the cell are synthesized. Number 3. Metabolic pathways are taking place with the help of sequential enzyme systems. These pathways are regulated at three levels. Through regulation through the action of allosteric enzyme, which increase or decrease the activity under the influence of effector molecules. B. The hormonal regulation. The hormones are chemical messengers secreted by different endocrine glands. C. Regulation at the DNA level. The concentration of the enzyme is changed by regulation at the level of synthesis of the enzyme. In this diagram, there are cortisol and insulin which act on skeletal muscles, liver, and adipose tissue. Cortisol increases gluconeogenesis, whereas insulin decreases gluconeogenesis. Coming to types of metabolic pathways. A. Catabolic or degradation pathways, where energy is rich complex macromolecules are degraded into a smaller molecules. Energy released during this process is trapped as chemical energy, usually as ATP. B. Anabolic or biosynthetic pathways. The cells synthesize complex molecules from simple precursors. This needs energy. C. Amphibolic pathways. These are seen at crossroads of metabolism, where both anabolic and catabolic pathways are linked. Coming to stages or phases of metabolism. The degradation of foodstuffs occurs in three stages. In the first stage, digestion in the gastrointestinal tract converts the macromolecules into small units. For example, proteins are digested to amino acids. This is called primary metabolism. Number two, then these products are absorbed and catabolized to smaller components and ultimately oxidized to CO2. The reducing equivalents are mainly generated in the mitochondria by the final common oxidative pathway or citric acid cycle. In this process, NADH and FADH2 are generated. This is called as secondary or intermediary metabolism. Third, then these equivalents enter into the electron transport chain or ETC or respiratory chain where energy is released. This is called tertiary metabolism or internal respiration or inner or circular respiration. In this diagram, we can see that there is primary metabolism or digestion of carbohydrates, lipids, and proteins into glucose, fatty acids, and amino acids. These undergo secondary metabolism, that is, it forms carbon dioxide inside the mitochondria. Later, they undergo tertiary metabolism, where NADH is taken and stored as ATP, that is chemical energy. Here ends our session. Kindly like, share and subscribe the channel. Thank you.